Hello and welcome to this AimHi Live. Thank you so much for coming. Today, this live is a science live, and this one is a very special one because we're bringing on an inspiring guest. So about a year ago, our next guest showed me some short videos that literally changed my life. I think there is a big difference between understanding something and feeling it. I always knew that there was loads of tiny life everywhere that we can't quite see. But it wasn't until I met our next guest that I really started to appreciate just how incredible and colourful this hidden world is. Dr. Sally Waring is a superstar biologist from New Zealand who's been researching everywhere from the universities of Melbourne to New York, has just completed a post at the American Museum of Natural History, and is now at the Earlham Institute here in the UK. She is a huge hit on Instagram with her page Pond Life, which you have to check out after this. And I'm really excited about what's about to come. Hi, Sally. You're in. Oh, Venerable Sugar's here as well. Hey, so what are we going to do today?
Today we're going to have a look at this little sample of water. This is a sample of water that I collected from a ditch, just a puddle on the side of the road near my house. But what we're going to do is put this little bit of water on a microscope and magnify it and take a look at all the living things that are living in this puddle on the side of the road. So we'll put the organisms on this microscope, which has an objective, which is a focusing lens with lots of pieces of glass in it. And what that's going to do is magnify whatever's on this slide. And then my camera is connected up to this microscope. So it's going to record what we can see. And then I can beam what's in my camera to my computer. And then that will be shown to you.
Amazing. So first, you sent me a video. This will give you the background of what's going on.
The other day, I was out walking and I saw a ditch filled with some very exciting looking water. So what we're going to do is head out and check out what kind of microbes we can find in that ditch. I'm going to add that to my jar. Now, you don't need much water if you want to look for pond life. Microbes are really small, really tiny, and you can find thousands, even millions, in a little sample.
So what I've done is I've put a little bit of that water on a microscope slide, and that's going to go into the microscope, and then I'm going to set up my camera. So let's just see what we can find.
Oh, here we go. We are ready. Whoa! What's this?
So we are now live in a puddle sample.
We are currently looking inside a puddle, just water in a puddle, and it's an absolute jungle in here. And Dr. Sally Waring is taking us through it.
We're just gonna have a little look around and see what we can see. There's something moving there. Do you see?
What's that? It's like
that is a organism called a ciliate. It's a single cellular organism, so that creature is just one cell big. This is actually a specific type of ciliate called a colex ciliate. It's a very charismatic little organism. It's a predator. They have kind of mouth parts. They aren't really mouth parts, but they act like teeth. And so the colex ciliate is able to latch on to a piece of algae, which is what it likes to eat, and then kind of suck the algae's insides out of the algae cell. It's really exciting to see it happen.
Whoa.
It eats and it hunts just like wolves or lions or any other large hunting animal, but it's very small and it's not an animal.
Yeah, how small is it?
This is magnified about 100 times. So the view that you can see on your screen, you can probably see about a millimeter there. So I guess it's about 100 microns, like 1/10 of a millimeter, I estimate, or maybe a bit smaller, maybe somewhere between 50 and 100 microns, I reckon.
Wow.
Now this organism here, this is quite a big organism. This is actually a multicellular organism. You see this one? This is actually a tiny microscopic animal. So even though it's not really much bigger than some of the single-celled things we've been seeing, this is a rotifer. It has a mouth, it has a stomach, it has a tiny little nervous system, so a few neurons together, which kind of form a very primitive nervous system and brain. Let's try and find. Let's maybe go up in magnification.
Ooh, what's that long thing? Whoa, is that the same thing we just saw before?
Yeah, that's another rotifer there. So it also has a little tail and a foot. It's not really a foot, but it acts as a foot. And with that foot, it kind of grabs onto the substrate and that allows it to stay anchored in place.
Wow.
This is a little algae called a ficus. You see, it looks like a little leaf, little falling leaf.
Wow.
It's a thing called a euglena. So it's a type of algae. And this one is called ficus. It's one of my favorite organisms to see just because it's so beautiful and graceful. It just kind of floats around in the water and falls just like a falling leaf.
Is it like powered by something?
At the front of its cell, it has a thing called a flagella, and that flagellum is kind of like similar to a cilia, right? So, but it's thinner and it's longer and it's slightly different structured, and it sticks out the front of the cell and it beats like a rotary motor, and it's able to pull the cell through the water, right? Things that are this small, water is much more viscous. It's like moving through honey. So these cells have to work quite hard to move, and so that flagellum pokes out the front and kind of moves around and pulls the cell through that water, that honey.
That's so interesting. I never really thought of that, but yeah, that makes so much sense. The water would be really hard to move through. It'd be like jelly. Wow. Why aren't these moving? Is another question coming in. Are they on a date?
Yeah, maybe they are. These actually do move. We'll hopefully find some moving a bit later. These are two other types of algae, the diatoms, and they're another type of algae. You can see that they're a different color from the phacus, so they have slightly different photosynthetic pigments. Wow. And they're both kind of shaped like boats, and that kind of structure around the outside of them is actually a glass shell. So it's a shell that the diatom has synthesized out of silica. It's called a frustule.
It's glass.
So, yeah, it's made out of glass. Yeah. Wow. So diatoms will collect silica from their environment and then extrude it around themselves to create this beautiful thing called a frustule, and they live inside that. Is that we've got a little green cell. This is actually another type of euglena. It's not a phacus, but it's related to the phacus. But can you see that there's a whole lot of other tiny little things around that?
Yeah.
Those little tiny moving things are all bacteria. So this organism is releasing a bit of oxygen. And these bacteria are able to sense that oxygen, and they like the oxygen, they want it. And so what they're doing is they're crowding that photosynthetic cell to get a little bit of that oxygen.
Wow, that's so good.
Okay, I wanted to show you this big long green thing here. This thing is also another type of bacteria. This is actually called a cyanobacterium. It's a big long filament. There's actually multiple cells in this filament. And you can see it's able to glide really slowly. It's moving a little bit. So this is the thing, if you've ever heard of kind of blue-green algae, right? So they're a different kind of group of organisms from some of the algae we've seen, but they're also photosynthetic. These little cyanobacteria are responsible for a lot of changes in our planet, right? So about 2 billion years ago, cyanobacteria became so abundant, and they did so much photosynthesis, and they released so much oxygen into the environment that it completely changed our environment.
You know how that's the thing where like parents will often allow like young kids to have a pet because when the pet dies, it helps us to learn to understand death, because obviously death's an important part of life. Do you think that everyone should have a pet cell so they can experience it?
It's not a bad idea. Though, they might want to have a pet population of cells, because cells themselves can, depending on the cell, depending where it is, sometimes they don't live for very long, right? So some of these cells, the fastest bacteria will divide once every 20 minutes, right? These organisms that we're looking at will be a bit slower than that.
Yeah.
They'll divide more like once every day to a couple of days. They do reproduce very quickly. Cells reproduce by dividing, right? So you start with one cell and it goes through mitosis and becomes 2 cells, right? And lots of people ask me, once that cell's gone through division and become 2 cells, are those now the same organism, or are they 2 new organisms, or has the first cell died, and are 2 new cells alive, or is it the same cell, just now 2? Like, it's kind of interesting questions to think about.
I've been reading about diatomaceous earth, and it's like a weird. People use it as like like an abrasive. So
diatomaceous earth, when a diatom dies, its frustule is left behind.
So that's the glass bit.
Those sink to the bottom of the water column, and they settle in the sediment, whether that's in the ocean or in fresh water.
So sorry, in simpler words, so the glass bit around the edge of the diatom is good. Yeah. It falls down to the bottom of the sea, and then you get a pile of the glass, like literal glass that's been made by living things.
That's right. Yeah. And that's what makes up diatomaceous earth. It's all those shells, or diatoms, just kind of built up over a really long period of time. This is an empty diatom frustule, right? So this is one of the things where the diatom cell has died and it's left behind its frustule, and that's the thing that'll fall into the sediment and then eventually become diatomaceous earth if there's enough diatoms in it. This one is also a euglena, but this one's a different species, and it lives inside a shell as well. So it like the diatoms, it also builds a shell. The shell's kind of around the outside, that kind of browner material, as its shell. It's called a theca. And you can actually just make out, I'm not sure if you can make it out, you can just see the flagellum poking out there and kind of moving around in the water. Inside the shell, there's actually the green euglena cell, which is sitting inside there. It's kind of a proteinaceous shell made of protein and minerals that are collected in it and exuded by the cell. You see there's a wee brown dot towards one end of the cell as well. Do you see that?
Yeah.
So that's a very primitive light sensing organelle inside the cell. So what that allows the cell to do is sense the direction and the intensity of sunlight.
Wow.
Right? So these cells can tell what direction the light's coming from, and it allows them to either swim towards or away from the light depending on how much light they need.
That's amazing. Wow. I can't believe these things have eyes.
Oh, here's a big diatom here that's actually moving.
Whoa.
So you can see how diatoms move. So there it is. There, that's the big diatom. And it's just kind of down one side of its frustule, it'll have a kind of a groove, and out through that groove, it excretes a mucilage.
What's a mucilage?
Mucilage. It's kind of a proteinaceous, gooey.
Wait, what's proteinaceous?
Lots of proteins in it. It has lots of proteins in it. It's kind of.
So a slime.
Yeah, it's
Cool.
a slime. It's out its slime, and then just kind of glides along its slime.
So it's like a tiny slug.
Yeah, it's kind of like a slug, like you see that trail of the slug, right?
Yeah.
Diatoms.
Ooh.
Oh, I need to see it. I was looking for earlier.
Ooh, what's that?
So if you just look closely at one end here, that's its kind of mouth. Coming out either side of that mouth are actually a bunch of cilia. So it's also a cilia. It's covered in cilia, but its cilia are arranged in a way so that they form a ring around the top of the cell. And then what happens is that those cilia beat back and forward and they create a current, and that current brings water into the mouth area of the cell, and it'll swallow up that water. And if there's any kind of bacteria or other small critters or small organic particles in there, it can eat them.
Wow.
Oh, this is a cool thing coming up here.
Whoa.
So what these cells have to do is just constantly pump out fresh water from these cells, so it's always rushing in. So they have this thing called a contractile vacuole, which just fills up with fresh water and then just pumps it out and it just runs all the time to keep the cell kind of osmotically balanced.
Why is that diatom carrying a shopping bag?
Yeah, it looks like it, doesn't it? Because they have that mucilage, that kind of mucus slime that they excrete, sometimes they do get stuff stuck to them, and you can see them dragging stuff around. I don't think they mean to. What else have we got? It's a funny little thing.
Whoa, what's that?
It might be a fungal hyphae. It might be a type of, here's a cool thing. It might be a type of bacterium I was just saying, but here's actually another bacteria here. Do you see this kind of ropey thing covering the screen now?
Wow.
This is actually a bacterium called a sulfur oxidizing bacteria. And it's also a filament, right? So there are multiple cells in this filament. And all those dark little spots along the filament are little sulfur granules that it's slipping off.
Wow. Would this water be safe to drink? And if not, why not?
Is this water safe to drink? Organisms that thrive in water don't always thrive in our stomachs, right? Because they're a different environment. There are things that do thrive in water bodies that will also thrive inside you, and that's a problem.
There's quite a big campaign at the moment going on with trying to get a bit more soil back into our diets, because apparently we don't get enough soil, and actually we need some of the little microorganisms that live in there to help to stimulate the right bacterial balance in our stomach. So, maybe a bit of pond water every now and then just before bed isn't too bad.
I'm not going to recommend that.
Okay.
I want to do the experiment and let me know the results.
Yeah, I also would not recommend that, but it's a turn of phrase, isn't it? So I was just going to say that I love it when we're just watching a scene like this when nothing's really happening, but then something just photo bombs across the top, really close to the camera, and looks massive.
There we go. So things kind of photobombing, those are the ciliates. They're really big ciliates called paramecium. These get so fast, they're really hard to keep up with. But sometimes you can, here, this one. Yeah.
Whoa!
Sometimes you see them doing funny things, and I swear there's some kind of social behavior going on.
If we were to take this kind of pond sample and we were to pop it on a planet where the water was stable, how much of this stuff do you reckon could actually stay alive?
Well, yeah, it would depend on the planet, right? It would depend on whether its kind of atmosphere is similar to ours. Bacteria would have the best chance, I think, of surviving. But, things like a lot of the organisms we're looking at, a lot of the larger organisms are dependent on oxygen. And the only reason, they've evolved in a world where there's lots of oxygen like ours. So if that changed, a lot of them wouldn't even survive on Earth.
Amazing. Right. Well, thank you so much for coming on. This has been so great. I've really, really enjoyed it. And I'd love to. I really hope we get to have you again sometime because this was amazing. But otherwise, here are the socials for AimHi, which please do follow those if you haven't already. And you can sign up for our schedule on our website if you haven't done that yet. That's our website, just above, just there.
Well, thank you very much for inviting me on.
Thanks for coming. Right. Farewell all. Goodbye. Ooh.